Hello, George, and Food Forest. I'm going to look at a compost I've had going for quite a while out front. This great big one. I've shared videos of me turning it. It's 141 degrees today. There's snow on it. I took a picture of that. But thought I'd take people with me to show the whole process if this is new to you. So I'm going to show how I take a sample of it real quick here. I've got it over here in a glass. I've mixed it in. Usually I'd be doing this at a set dilution so I can compare it to other samples so I'd know what proportion water to what proportion compost. Right now, we're just wanting to have a look through and see. I'm going to put one. I can even do 2 drops of water if I want, but I just want to pay attention to that. We'll just go with one. Just so that it's a single drop of water. I got to try to do this without hitting the camera. What I'll do is I'll make the slide touch it, drag it backwards a little bit, and drop it so I try to get an even coverage. There we can see it was one tiny drop of water. I did pretty good, probably 3/4 of the slide. I could have gotten better coverage over here. Sometimes by tapping it a little. I'll pause here and put it under the microscope. All right. What do we see here? So we got a nice chunk of fungi in here. That might be a piece of root, actually. Take a little better look. Because it's not uniform. And we're seeing branching that's not uniform. It gets thicker to thinner. We know that's a nice piece of root. I may pause at some point and use a more directional light on that, and we'll have a look. What else? We'll try and find a nice piece of fungi to compare it to that. There's one. It's not a great one, but see if we see some septa in that. Let's find a better one. So like that's probably going to be an amino acid. There's a piece of fungi in there. We'll go all the way into 400x. Maybe get a little more contrasting. We actually give it more light. Here we see the septa, the breaks in that piece of fungi. I'm pretty sure it's going to be a saprophytic fungi, a decomposing fungi. It's a beneficial, that's what we want. We saw the bacteria moving around in here. This is a hot compost, a thermophilic compost. So it's going to be bacteria heavy. And we see there's a lot of bacterial movement in there. I do believe I see a flagellate down here. Oh, look at that fungal spore right there. There's a fungal spore with a flagellate next to it. Pretty sure we're going to see a flagellate there. They're usually on an outer level like this. Hard to really make them out just from the motion and the spiraling, I believe it is. And this here is a fungal spore. Another good sign. There can be bad fungal spores as well. Candida can be a double-ended spore like that, I believe. I'm just learning more about that. There's another nice spore. Being a wood chip pile breaking down, it should be fungal heavy. It's been going for 6 months now. We're close to it. I keep adding more food to it when I get drop-offs from the local food waste, food scraps. I believe that's going to be a flagellate as well. You can't make out the tail on it, the flagellum. I'm doing this to teach others, and if anyone else sees things that I'm misspeaking on and needs to go more deeply, please comment. That kidney bean shape, I believe that's a fungal spore as well. I'm not going to say. I think that's lacto. Could be wrong though. Lactobacillus. Don't take that too much. Look at all those fungal spores there. I believe we're also seeing a testate amoeba. That just means a shelled amoeba. We can see the 2 cell walls are on the outside of it. Some nice fungal spores. Some fungal hyphae we'll see in there as well. Some broken up pieces of the branching of the fungal hyphae. Let's back off. We'll go to 100x. Just do a little look around. Who's that? Things we'll see in here will be called cysts sometimes. They'll be round like this. And that's one of these animals. One of these, I don't believe that is. I'm not sure what that is. Maybe it is. And there'll be one of these animals in hibernation, whether it's a protozoa, could be a nematode egg, uh, amoeba, gone to cyst, gone to sleep, gone to hibernation. There's a good looking flagellate right there. We can see all the motion. We can make out his tail a little bit. For a long time, I was wondering what flagellates were, and mostly by movement. And I was calling a lot of bacteria flagellates, I believe. That's a pretty good shot of one there. Let's contrast more, whatever it's called. Put a lot of light through them and have a look. Yeah, that's not helping. But that's one of the upper trophic levels. So we saw the shelled amoeba, the testate amoeba, and this flagellate. And they're going to be that second trophic level above bacteria and fungi. All the little things floating around are bacteria. We've been talking about the fungi we've seen. So that flagellate feeds on them, and so would those amoeba. So the nutrient cyclers for us. And back off one more time. We're at 100x now. There's a nice looking amoeba, I believe, shelled amoeba, right here. Get a good look at him. We can even see his insides there. The oval shape, one flat spot on the end. That'll be the mouth. As I go in and out a little bit, you can see the two cell walls, the testate they're in. There's also just regular amoeba that are like blobs moving around. They're a little harder to see. Sometimes I wonder if I'm seeing them or not. They're so indistinct sometimes. Like that right there. I think that's just a mineral, but it looked like one. Let's back off again. Maybe we'll go out to 100x. I'm sorry, 40x. And just do a general search around a little bit. There's a nice looking piece of fungi. Let's have a look at that. We'll go into 100 first. As I go in and out a little bit, you can see the, excuse me, the hyphae on here, the branches of it, each of those being its own individual chamber with cytoplasm flowing through it and carrying nutrients and proteins. And I'm just reading Teeming with series, and it's just fascinating all the things that go on, the interactions. This is life. This is the base of life here. We can see a fungal spore and a fungal branching hyphae, like the root. It carries nutrient through. Actually, this is a decomposer. I'm not going to say too much because I'll say it wrong, and I'm still learning so much. How long is this video now? Oh, we're looking at 10 minutes. I think I'll probably stop there. Is there anything we haven't seen? We haven't seen any nematodes. I've showed those in other videos. But we've seen some of the protozoa, and those were the shelled amoeba, the testate amoeba, and the flagellate. And this right here, I think it's a flagellate. I thought it might have been ciliate, which we don't want to see too many of those, but it's a flagellate. There he is, bumping around, bumbling around. I've lost him. We'll see things like this most likely in the yellow color. And that's going to be an acid. That's going to be not humic, but fulvic acid, I believe. And fulvic is just on its way to being humic acid, I believe, in a very general state, very general way of saying it. We'll do a little look around here, about 10 minutes long. Anyone's watching, I appreciate you watching. I'm just trying to teach as I learn. So again, if anyone's watching this and saying bullshit, that's not, please comment. But I think I've gotten it done pretty well now. I haven't taken the class, but I've talked with others and been looking around quite a bit. Sometimes I'm confused. I believe that's a shelled amoeba, but I'm not sure. I don't see any hairs on the outside, which would make it a ciliate. It might be a flagellate. I can't see the flagellum, but there's a chance it's a flagellate. I don't really see the mouth to make it a amoeba, but I think it is amoeba or flagellate. These fungal spore, all little dots moving around. If I pull in, oh, there's another flagellate, I believe. If I pull in those dots, we'll see a little different color to them all. Greens and reds and blues. Those are bacteria. And the different movements within them. Some are going to be rod bacteria. There'll be a simple straight line, a rod. And cocci bacteria, little dots, bacilli. And then there's going to be spiral ones, which look like a Z, a zigzag, and we don't want to see those. Or too many of those. So I believe the terms would be cocci, bacilli, and spirilla. 3 types of bacteria we'll see. And hopefully we're only seeing the first 2 in here, cocci and bacilli. Yeah, I'm messing that up a little bit. Google it. Oh, what else? See, like these are probably cysts, I believe, something that's balled up, hibernating. There's a good looking testate amoeba. You can see the shell in the mouth and the insides. There might be another one over there. We're just going to go real quick, see if we see anything exciting. Lots of, again, this is a wood chips. They take a long time to break down. So we're going to see a lot of these fungal spores in there. There's a fungi that's in there, sporulates, and more of them feed and reproduce. Different ones at different temperatures for the bacteria. I think we'll probably end here unless I see something more. I'd like to see a nice fulvic acid, a nice, more golden piece. Maybe we'll back off one more time and have a look. Minerals as well. The more shiny things we're seeing are going to be minerals, I believe. So we're seeing aggregates, minerals, bacteria, fungi, protozoa, fungal spores, fungal hyphae. We're not seeing any nematodes in here. We see a lot of those in the worm compost. This would be plant matter, organic matter of some sort. Some nice fungal hyphae flowing through it and around it. But pretty interesting, anyways. Excuse me. For a single drop of water, as we saw at the beginning, this is so much going on in this single drop of water. We'll go in one more time. We'll go all the way out from there. So we're 400x here. We're seeing a piece of plant material and the individual cells within it, I believe. We'll go off of it a little bit and see that fungi, that fungal hyphae next to it, possibly even going into it. You see all the bacteria in the background if we zoom in there a little bit. Come away from it a little bit down here, bring it a better view. And you can really see the bacteria in the background and that fungal spore. All the different colors, shapes, sizes, and motions. Sure, really don't know exactly what those spores are. We're seeing a lot of this spore here. There's three in that same view, but that one. If anyone knows, I'd love to know. Thanks. Oh, geez. I'm trying to take a picture over here. All right, I'm George. Oh, I said we go all the way out. So here's 400x out to 100x. There's that testate amoeba and the fungal spores all over the place. Another amoeba, I believe. Plant material. And out to 40x. One quick zip at 40x. All right, I'm George. Fat Ninja Food Forest. There's a big testate amoeba. I'm George. Actually, we're going to be having a microscope class here. So I share this today, Saturday. This is supposed to be the compost class. We're going to have a microscope get together here tomorrow, Sunday, the 8th, at 3 p.m. So, chance to be better than a holiday. Peace.